Oh, okay. Just like
you. It's recording now. All right, welcome everyone to the Tools and Technology Seminar Series. That's a great turnout, so it's very glad you guys to come. I do see a few new faces, so if you haven't been to the seminar series before, do my little spiel. It's just basically an opportunity to discuss and learn about new tools, technologies, methodologies, and new tools, because they might be new, they might currently be in development, they may be something that's been around for a long time, it's just been used in a novel way, or may not be aware of. So there is a sign-in sheet. I think most people signed in. It's sitting up front. Actually, I'll just take it to the back. But if you haven't signed in, please do so. It just helps us justify the pizza. Just proves that we had people here to eat it, which is always a good thing. So we want to continue offering pizza. So that, I'll just introduce our speaker. So today's speaker is Cristina Mitrea, and she got her PhD in computer science with a focus in bioinformatics from Wayne State, and is currently a lecturer here in DCMB.
Yeah, and today I'm going to talk about something a bit different, which is qualitative change detection approach for preventive therapy. So this is a paradigm shift from most approaches tend to find treatments and look for treatments. My approach is trying to find ways to prevent the disease and do preventive steps towards not towards keeping the healthy state. So currently, more diseases are diagnosed after significant disease-associated changes have occurred. So this typically happens when people are trying to look at data from disease and try to find treatments for it. We propose a paradigm shift for keeping the healthy state. We're still going to use data that has treatment and control, but in this case, we're trying to find when the shift happens from normal to disease to be able to keep the normal state. So the framework is that we have a lot of data that's coming in. There are high throughput technologies that are being developed, and more data is being collected, and more high dimensional data is being collected. So we have a lot of complex data available. And there is a challenge because technology is not keeping up to extract the knowledge from that data. And a typical high throughput experiment contains two different phenotypes, contains control and condition, disease and healthy, and it extracts either a set of genes or a set of pathways from there. So we have parts of a set of genes that have interactions between them. So we have gene parts, gene sets. We have sensor reads, which is the expression data, and this is more of a engineering technical look at how we can interpret expression data and gene sets. And we have another dimension that typically is not included when you do pathway analysis, which is time series. When you are trying to identify systems that don't work, typically don't look at the time. So methods that look at part sets usually find patterns. Methods that look at the data that comes from the sensor from gene expression typically find what is different. They do enrichment analysis. They find what is different. What elements are different. We can find patterns. We can find new blueprints, new pathways, new networks, and we can find differences using those blueprints and networks, which is enrichment analysis plus topology, which is pathway analysis. So we can account for the interaction between those parts, which typically the part set, the gene sets don't. And my research, my interest was lying in between finding differences using those blueprints, using the topology, and also using time in this analysis. And this is called the systems approach because it looks at the whole system instead of looking at just one gene at a time and saying, is this different and then is this different? Typically, genes act in tandem. They don't act in isolation. So that's why a systems approach is a good approach. It looks also at emergent properties of the system that might not be available if you just look at one part. The example that I always give is we have a car. The car has the different parts. Unless they're put together, the car doesn't move. So that's an emergent property that you can have only if the system is put together. So looking at the system's view, like a blueprint of the engine of a car, we can identify what's wrong in the system. And those blueprints, like the map of an engine of a car, are biological pathways. And we have different types of pathways that are available, that have been constructed using knowledge that has been developed. We have signaling pathways where genes interact with each other. We have metabolic pathways where we have a compound that gets transformed in another compound, and that reaction is being catalyzed, is being sped up by an enzyme. We have different type of pathways where we have different types of nodes, like component nodes and process nodes, which are in the NCID, the NCI, the National Cancer Institute pathway interaction database. And we have these type of structures because we want to store this knowledge that we gain from doing experiments and identifying interactions between them. We also have directed interactions and directed interactions and undirected interactions. So we have a lot of complexity in the pathways too. And that's why pathway analysis is important, telling us which part of the system doesn't work. Pathways typically describes specific processes that might be disrupted in a condition. So use this to gain insight into the disease or to gain insight into the treatment of the drug. And how we do that? We do an analysis that ranks the pathway, identifying which are the most disrupted ones in that condition. So typically, a pathway analysis tools takes information like molecular interaction knowledge bases, pathways, and biological experiment data, gene expression, methylation, protein expression. It does the pre-processing of the input data. It does mathematical and statistical modeling to score the genes, and then to score the pathways, or to score the proteins, and then score the pathways. And then, after the pathway scoring is done, we have either a significance assigned to that score or just the score, and we rank those pathways. So that's the typical process for a pathway analysis. But nowhere here we are using time, and I think time is a very important dimension to integrate in such an analysis. There have been a lot of, this is a bit of an overview of the methods that have been developed so far for pathway analysis, and there have been methods developed for metabolic pathway analysis, much fewer than the ones developed for signaling pathway analysis, and none of them are using time. So here there is a different view of the same methods. They are categorized by the type of scoring that they do, aggregate scoring, linear, nonlinear. That's the scoring for the pathway level, and we have node scoring level, which is the gene or the protein level scoring, which can be used using graph measures, using similarity, like correlation, using probability approaches, or using normalized gene value or node value. So we have a lot of approaches that have been developed, and yet none of them include time. So that's why this is an even better overview where we have pathway analysis methods that are more than 30 that have been developed, gene set analysis methods where the topology, the interactions between genes, as you see here, are not considered. We have even more of those. We have methods that infer networks that discover new relations between genes. And we have only 4 methods available that actually try to consider time in a way or another. So that's the scope of this research, is to develop a good method for integrating this type of data and extracting knowledge and answering the question, when does that change from normal to disease happen? When does that change from the effect of the drug happens in the organism? And I name it technical qualitative changes in biological system, and it's QCD. That's an abbreviation of it. So the motivation was to identify systemic transitions between states of the system. So we have an organism doesn't go from normal to disease in a rapid progression. For diseases like Alzheimer or neurodegenerative diseases, it can take a long time, until the disease manifests. But changes happen behind the scenes, and changes are accumulating until a potentially irreversible change occurs. For instance, I have an example here for liver damage. We have healthy liver, damaged liver, and then we can get to liver cancer if no intervention is being done. And this is similar to the idea of wearing a fitness tracker, for instance, that can track your heart rate, that can track different inputs from your body. They can track how well you sleep. They can, at some point, maybe even track sweat content, and they can identify if changes happen in a certain direction, and they might actually allow you to take steps into preventing those changes to progress to the disease. So that's the overarching goal. The method that can be used to detect these systemic changes. And these changes, if you look from one single time point to another single time point, they might not be readily available because the change is progressive. So either you see the change when it's already at the disease state, or you don't see the change because it's between two states that are too close to each other. So the application is identifying disease before the phenotype is present. The analysis context is to the detection of change intervals uses pathways and sequential data, uses those blueprints, the gene networks, and uses time course data because it tries to identify an interval of time. And why is this? It can use different types of sequential data. It can use neuron signals. It can use disease progression, and that's not time course, but it's still sequential data. If you progress from one stage of the disease to another, it outputs change intervals, and it's designed to identify when that change happens. And existing methods so far are not suitable for this, and that's a bit of related work so that we know there is a need for this method. Supervised machine learning, they have no labels. Supervised machine learning, there are no labels in this case. We don't have the disease and no disease labels for this kind of data. To use enrichment analysis, there is no reference gene set in this case because we have just time course. To do pathway analysis, regular pathway analysis, there is no phenotype comparison. Again, there are no labels to compare. So how would we do this? Existing methods are not suitable as they are to answer this question. So what we thought of using is one of the existing methods and incorporating that time course information into it. So how we did that, we take experiment data, which are the different times or different sequences of the disease, and we take the system information. And for most organisms, we do have a lot of pathways available. We have pathways that describe some of the processes in that organism and in those cells. And we use that as a guideline to the interactions that happen between the components of the system. We compare every 2 time points. So we still have to do a comparison, but we don't have a reference. We can't compare just with time 0 because we don't know if that's where the disease starts. We can compare with time 0 because we don't even know if that's the normal state. So we just make all the comparisons because the goal is to detect if a change happened during the time that we captured this data. And after we do all the comparisons, we evaluate if the system, our system that's given by a pathway, by a network, has changed during those 2 time points. And we can do that with any pathway analysis method because it does compare to phenotypes from time 0 and time 1, for instance, from time 0 and time 6, from time 6 and time 2. So it compares every 2 time points exhaustively and computes that perturbation, that change, evaluates the change in the system. And they are assign a perturbation value, all arcs or changes between every 2 time points are evaluated. Then we select time intervals with large perturbation that are changed, and those will be the red ones. We mark them. And we select that using a gamma mixture model, and I'll detail that later. And once we selected those intervals that changed, we have to narrow it down. It might be that we have overlapping intervals that change. So we need to identify the narrowest interval where the change might have happened. And these intervals reinforce each other, for instance. They can reinforce each other, and that means the change happened between here and the very next point where we have a red arc. I'm going through more details with that. So once we narrow it down, we have the list of change intervals, and that's the goal of the method. The novelty of the method is this part, to compute that narrowest interval. Because any pattern analysis method could be used to actually compare every 2 time points. But how do you identify the change interval? You will end up with a lot of these kind of values, which you won't know what to do to evaluate which is the interval that is of interest to you. So we run this method on multiple data sets. We run it on simulation data. We run it on real gene expression data. We wanted to see how it behaves. Usually when we develop a method, we want to evaluate it, and we want to compare it with other methods to see if it does better. So we compared it on synthetic data for E. coli flagellum building. We used a simple organism, model organism, because it's very well studied. It has a lot of networks that we know of. The system has all the parameters kind of worked out. So it was good evaluation candidate. So when in an environment lacking nutrients, E. coli builds a flagellum to help it go to an environment that has nutrients. And that's the phenomenon that we're simulating here. We have the flagellum building network, which is our system, and that's been presented in literature previously. And we generated data for 10 hours, sampled every 10 minutes for a total of 21 time points. And this was implemented in R. So I wrote code to actually generate this data. And the known change interval, because the phenomenon is so well studied, we already have that answer. Now we're going to run our method and see if that matches the expected answer. Is the network from literature, and these are thresholds when the gene expression of the next gene starts accumulating. When the next gene starts being expressed, the next group of genes starts being expressed. We have a master regulator. We have another regulator, and then we have 6 groups of genes that are being turned on by those regulators. So we chose a simple network so we can control how it behaves, and we can properly evaluate our method in a controlled environment. So to simulate the data, we use the functions for protein accumulation and decay. And we use the parameters, as I mentioned here, from previous literature to actually generate that data. We displayed the data, and we saw, because it's a controlled environment, it shows very clearly that there is a change here. There is a shift from low expression to high expression. And if you think about it, this first starts activating this gene first, and then this group of genes, and then this gene, and together they regulate more genes and more genes and more genes. So the expression accumulates for all genes. And here on the y-axis we have the genes from the network, and on the x-axis we have the time in minutes. And here I displayed the depiction of the flagellum being built. Yes.
Usually goes 20 to 30 minutes per generation. So does your data suggest that if an E. coli wants to grow a flagellum, it needs several generations growing.
So this is scaled data.
What
Is scaled. The data is scaled. So
do you
we
mean by scale?
scaled. We reduced the time scale. This is simulation data.
No matter whether it's simulation
Okay.
or not. So does your results suggest that E. coli needs several generations to
No, it doesn't.
So if it doesn't, then why is this correct representation of
So here we were trying to show at a different scale how the phenomenon is happening because it's simulated data.
Yeah, but the phenomenon that's happening should not be that long.
Shaxin, think about it this way: if the E. coli grows at like 37 degrees in LB, it's about 20 to 30 minutes, like you said. But the phenotype Cristina is looking at now is in a nutrient depleted condition. So I don't think they would be able to grow 20 to 30 minutes that fast.
That means in such environment, they probably grow much slower.
Yeah.
And
that's why the data is scaled.
Is around the time they duplicate.
I guess it depends on the exact environment, but I think a 30 minutes time interval is pretty reasonable.
So it's scale data. It's not. We use the parameters from literature and we generated the data. We didn't take data generated from experiments in this case. So this is not. It doesn't replicate exactly the data from experiments. So what we do in this case, we compare all the system states using a pathway impact analysis. We chose this method to do the comparison because it takes into consideration the interactions between genes. For instance, if a gene that is changed affects a lot of other genes, that's more of a regulator, master regulator. So we want to give that gene a higher weight. So we propagate the change that happened to that gene to the downstream genes. And in the case where a gene is lower on the hierarchy, on the pathway hierarchy, then it gets this change gets propagated only to lower levels. So it doesn't affect as many genes as it would be. A affects B and E and F, and through that it affects D and C as well. But in this case, because B and F are affected, only B, F, D, and C are affected. And this is an example of the perturbation factor computed in the two cases. So when we have this change, and the changes are similar, we have actually here the change is greater. We have a 7 perturbation factor, and here even the change is smaller. Yeah, change is smaller. We have a higher perturbation factor. So the weight of the genes, the way the genes are considered, and the way the change of the gene is projected or sent downstream, it helps us in evaluating better the pathway and the change that happens in the pathway. In step 2, and there are, as I showed before in the timeline, and as I described in the pathway analysis methods, there are a lot of methods that do pathway analysis, but this is one of the few that actually considers topology in this way. And that's why we chose this method. To select intervals with a large system perturbation, we take the distribution of those perturbation factors, of those changes between every 2 time point, and we fit 2 gamma distributions to it, because we expect to be comparisons with no change, and we expect there to be comparisons with high change. And that's how we want to distinguish between the comparisons with low and with high change. And once we identify those 2 distributions, and we fit those that mixture of 2 gamma distributions, at the intersection, we consider that the threshold between the high and low changes. And we put a 5% threshold if one of the distributions contributes less than 5% to the mixture, then we don't have a change. And that's how we evaluate. That means, and typically the high distribution contributes less in the case when there is no change, because we won't have high perturbation factors. To actually account for that, I can go back to the perturbation factor and explain a little more. So this is a positive value. It is computed from the difference in expression for each gene in absolute value that it's propagated downstream. So it's the perturbation factor, it's always a positive value, and the higher it is, the higher the change in the pathway. And that is why when we are modeling the distributions, we are looking for the high perturbation factors that will tell us there is systemic change, and the low perturbation factors will tell us there is no systemic. So once we did this, we wanted to know if that fit of the distributions is good. So that was a concern of ours. And we used different measures, like callback labor divergence is a measure of how a probability distribution is different from another one. We used the Kolmogorov-Smirnov test that quantifies the distance between the empirical distribution functions of two samples. So we used different measures that are complementary to see if our distribution was well fitted. Our two distributions were well fitted. And as I show a little bit further in the results, those gave us confidence when we looked at the results to either move on with the method or adjust it if needed to perform better. So after we identify the high and low changes, like in this case, we have large perturbation, and this is the example for the E. coli flagellate building for our simulation data. We have large perturbation, and we have small perturbations. And we can see that our large perturbations reinforce each other to the point that we can identify states that are before our change interval and after our change interval. So a change interval kind of emerges as the narrowest interval that has overlapping large perturbations intuitively. So there is a more formal description of this, but intuitively we are looking for the interval that has no changes in it and many changes that come before it and go after it. Any questions so far? The method. I lost everybody. Or you all got it? I think it will become a bit clearer as we go through. So this is the formal description. It basically tells you that we are looking at everything that's before the change interval and everything that's after, and we shouldn't have any change in the interval. And so it says is the maximum value to satisfy that condition, meaning this is the maximum value for this part, and this should be the maximum value of these states, and this should be the minimum value of these states. So nothing goes before this. So for S6, S8, or S7, nothing goes in, and nothing comes from it. They're all non-significant changes. So this algorithm was implemented in R, and once we ran our algorithm, we identified the change, and it coincided closely with the real change interval. So our change interval was detected between 180 minutes and 300 minutes, and the real change interval that was known was between 240 minutes and 270 minutes. So that means our change interval is a bit broader, but it contains the real change. Maybe with if we had more sampling, or if we used maybe if we had closer sampling, we would have gotten closer to the change interval. So after we identify the change interval, as we see here, these states kind of group together, and that gave us an idea and the thought of a meta-state. What if this is a meta-state of non-disease or non-effect of the treatment? And what if this is a meta-state of the disease or the effect of the treatment? So we tried to see, to evaluate if these are real meta-states, if this happens just by chance, or these are real occurring events in this data. And to do that, we took those groups of consecutive states before and after the change interval. From that, and all comparisons between states within a metastate have a small perturbation, and all comparisons between states of a metastate to states outside it have a large system perturbation. So again, we can see this here. So everything as before, everything that's between these states has a low perturbation. Everything that's between these states has a low perturbation. And everything that's from here to here has a high one. So that's how we defined the metastates. Because that gave us the opportunity to see if this is real. In the ideal case, we would have exactly this situation. Exactly this situation. And this is the ideal case. Everything fits perfectly. In the case of our data, it didn't fit perfectly. There were some changes that in the ideal case, when everything fit the definition of the metastate, would have had to be large perturbations, and they weren't, which is expected. Our method is not perfect, and we didn't expect it to be perfect. We expect it to give us good results that can help us identify that interval and help us identify metastates. To validate this metastate, we did a comparison with the ideal case, and we assigned to each of these comparisons a label. Is it consistent with the ideal case, or is not consistent? So if that number of consistent and inconsistent states, that under the null hypothesis, when there is no metastate, should follow a binomial distribution. And we use that. We did a test to get the p-value and to identify if those are real metastates, and that's the test we did. We compared it with ideal, and we evaluated if that happens just by chance, or if it's something that it's specific to the data. And for each of the methods that I used, or for each of the part that I'm describing, that's a computational part. I also put the package or the way that we implemented that algorithm. So we have our ideal case, we have our observed case, and for each of the metastate, for metastate 1, we compute how many are consistent and how many are inconsistent. For instance, these that are black are inconsistent with the ideal state. And do we expect that many to be inconsistent? More or less in the random case. And in the random case, we should the probability that a comparison is consistent or inconsistent should be 0.5, should be 50%, like rolling a dice, a coin, not a dice, like flipping a coin. So that's what in the random case, they're assigned random. They should have the same amount of consistent and inconsistent arcs or comparisons, and this is not the case. And for metastate 2, we did the same. And this is an overall view of all the evaluation that we did. So we fitted the distribution. We had the measures, the call-by-flavor divergence. We had the Kolmogorov-Smirnov test. We had the overlap, which tells us how good the area between the black thick line overlaps with the purple line. So that was a measure to see how the two distributions compare. The black one is the one from our measured perturbation factors. The purple one is the combination of our mixture model that we fitted to that distribution. And then for each potential metastate, we got a p-value based on the binomial distribution. So taking all this together, for the E. coli flagellum building, we have a relatively high overlap. We have a low call-by-table divergence, which we want it to be low. That tells us the distributions are similar. We have a high p-value for the Kolmogorov-Smirnov test. That tells us, again, that they're similar. We have more than 5% on our distribution of large perturbations. So that also gave us confidence that there is a real change. So overall, in this evaluation, we concluded that there are real metastates, and we do have a change that overlapped with the real change. We did this for real data too. So that was a case of simulation data, and we have more cases. I'll show a summary of the results. So we used biological experiment data for fruit fly metamorphosis. So the fruit fly goes through 3 major developmental stages. Can be a larva, pupa, and before becoming egg, larva, and pupa before becoming an adult fly. And that's a depiction of it. So we used that type, the data from the developmental stages, and then we used a hedgehog signaling pathway from a pathway database, which is called Kyoto Encyclopedia of Genes and Genomes. And we chose this pathway because it has a crucial role in organizing the body plan for the fruit fly during development. So that is a process that is relevant for our phenomenon. We took the gene expression from a public repository of gene expression data, gene expression omnibus, and we have the idea of the data set here. And also here we had a known change interval. Fruit fly is again a very well studied organism, so we knew when the change happened. And this study was published. It has a paper attached to it. Here's the depiction of the hedgehog signaling pathway, and the genes marked in red are the ones that were changed. So quite a few of them changed during the process, which was expected. And here we have the data in a heat map similar to the one that we had for the simulated data. We can see that this is more noisy. This is real gene expression data. It's not the controlled, simulated, nice-looking heat map that we had before. So we wanted to evaluate if our method actually is useful in a real case. And in this case, the real change interval was from -4 hours to 0 hours. And our method detected the interval from -18 to 0 hours. Again, it was overlapping with the change interval, and it included it. And even more so, the center of our change interval was lower than the center of the change interval, the real change interval. So we detected it earlier, we can say. But this evaluation gave us confidence. We wanted to see, is there another method out there that's doing something similar, and we should compare with that method and see if we do better, or if we at least do as good as they do. There is a method that we abbreviated DNBM, method developed to detect network biomarkers and the pre-disease state. Notably, this method doesn't identify change interval. It identifies only a pre-disease state, only one time point. As the input high throughput data and a large network of protein-protein interaction. That's another downside of this method, because processing such a high amount of data is very time consuming and resource consuming. So if we can do just as good or better with a smaller network, that would be beneficial. And the hypothesis for this method is that the leading network, so the network that they identify a small network from the large network of protein-protein interaction, and that network is the first to change towards the disease state. So that's the network biomarker that they're looking for. So what DNBM does is model the change in gene expression over time as a Markov process. It doesn't do the impact analysis that we do, where we propagate the change of the gene. It uses a different approach and uses a state transitional based local network entropy that quantifies the change in state. So it basically evaluates the network, again, a different state, but it compares always with the state 0. It doesn't compare all the states. It compares with state 0. So they do have a reference. They do set a reference state. Also, it identifies one single pre-disease state. It says this is where the inflection point kind of is. It doesn't identify the change interval. And we hypothesized that in our change interval, you could still kind of keep the healthy state. You could still reverse the changes that started. The evaluation of the NBM was done through functional analysis using experimental data and pre-existing knowledge. So they used gene ontology analysis. They used known processes and molecular functions that were related to the result to evaluate, to see if their result made sense, and they did. And also they used pre-existing knowledge related to the condition under study, which is also available information. You could use that. They did the study for 2 phenomena: mouse gene exposure and human hepatitis C virus induced hepatocellular carcinoma, so HCV to ACC transition. And we decided to do the analysis on the same data sets to see how we compare with them. In the human liver cancer progression, the infection with hepatitis C can lead to major complications, can lead to cancer, and can lead to liver failure. So in order to study this phenomena, we chose a relevant network for it, the viral carcinogenesis pathway. And this describes the signaling mechanism of an inflammatory response. So that it is relevant to see if that system changes. And then we use gene expression data for hepatitis C virus-induced hepatocellular carcinoma for 8 disease stages. And this is again publicly available in the gene expression omnibus. And here is a depiction of how you would go through those 8 stages, from healthy cirrhosis to high-grade dysplastic nodules to very early hepatocellular carcinoma up to very advanced. These are the stages that they had in the data set. The NBM, the previous data set, and now in this case we don't know when the systemic change actually happens, because this is a complex phenomenon and we have no reference. We don't know the ground truth. And for that reason, we used the comparison with the existing method, and the existing method detected a pre-disease state as very early hepatocellular carcinoma, which is very good. You want to catch the cancer early. This is a depiction of the pathway from KED, and the red genes again are the genes that changed in that experiment. And here, they detected the very early SCC as a disease stage. Our method, the QCD, detected the change interval starting from control to high-grade dysplastic. So it was an earlier interval, a quite wide one at that, but it was an earlier interval that allowed us to detect the transition from normal to disease earlier. So we had more phenomena that we went through and that we did the validation on. We didn't want to just limit ourselves to 3 or 4 phenomena. We wanted to make sure that this works on multiple data sets. And before I go to the results, any questions that you have about the method and the validation? Yes.
On your previous slide, your interval actually included the controls. Do you have a comment about that?
Yes, because this is the start of the interval. So it actually starts from the normal state. Yeah.
So it captures even the control's normal state.
Yes. And probably the control and cirrhosis were too close, so the change wasn't big enough to go to the next one. So it it's conservative in the sense that it finds a wider interval. Thank you. Any other questions about the method? So the intuition is we're trying to do an analysis that is not biased by reference, and an analysis that is exhaustive in the sense that it compares all states. And the challenge was, how do we identify that change interval? And we solved that by overlapping those comparisons that have large perturbations and narrowing them down. So the results show here that we had 8 phenomena, 1, 2, 3, 4, 5, 6, 7 model organisms, and we included in our validation two phenomena from the same model organism. For fruit fly, we included variation and acute ethanol exposure. We had a wide range of phenomena from some that shouldn't have a big change, like the avoidance reflex, but our method even captured that because all the other data points allowed for it to capture that. We had more complex phenomena like acute ethanol exposure, like fortune exposure in mouse, and the liver disease stages for human. So we try to have a wide range of validation to make sure that our method performs well and its general purpose. And for each dataset, we marked these data sources. If the data source is a mathematical model, that means it was synthetic data. We generated that to have controlled data that we can actually evaluate our method on. And then we had 5 data sets where it was a more noisy data set coming from real gene expression data. Here we have the detected change interval, the real change interval. In the case of the Bacillus subtilis sporulation, they actually matched perfectly. In most of the cases, they overlapped, and the detected change interval included the real change interval. In these 2 cases, we didn't have a real change interval, and this was what the method that was already published and did something towards identifying a pre-disease state identified. Again, that's why they have the card that denotes the results of PNP. We also validated our metastates for each of these cases. We wanted to see, do we really have the transition from one big metastate to another? For instance, in the E. coli case, we had from no flagellum to flagellum. In the hepatocellular carcinoma case, we had from no disease to disease. In the mouse carbonyl chloride exposure, and this is post-gen gas. That's very toxic. So in that case, wanted to see when the first changes in the mouse happens before even the symptoms or the physical symptoms that you could see happen. So the same thing for yeast sporulation. So we have a wide range of data sets. In the case of the metastates, for our control data, for our simulation data, they were all significant, highly significant, and they identified real metastates, which was expected. We had our data modeled using parameters for this phenomena and no noise. So for the real data sets, where we took the data sets from a public repository, we had 3 cases in which that particular metastate was not significant in the validation. For the yeast sporulation, that was the case. And for mouse carbonyl chloride exposure, in this case, we think that the change happened very quickly because that gas acts quickly. In the yeast sporulation, again, that was a wide phenomenon. So again, this is close to significance, but it wasn't significant. So we think that this might be a case of noise in the data that didn't allow us to identify that. and the fruit fly alcohol exposure. In this case, we actually have fruit flies that are exposed to ethanol, and then that state reverses. So kind of they get drunk and then they get sober. we have a state. They go from sober to drunk to sober, so it kind of reverses the meta state. And that's a special case too that we tested our method on. So overall, with these results, we felt pretty confident that we have a good method and we can use it on data sets that don't have a known change interval to identify it. There are, though, limitations to it, and that's future work that can be done for different diseases. There are limitations. This is implemented only for signaling networks. This is evaluated only with gene expression data. And it needs that system, that blueprint. So to address this limitation for needing a blueprint, most of the organisms do have quite a few blueprints available for them. But this is still a limitation because tying it to a blueprint might introduce bias. Evaluated only on gene expression data. We used in the simulation neuron signaling, and it performed well. We didn't do that on a real data set. We couldn't find one that had this kind of data. So we could evaluate it on protein measures. We could evaluate it on methylation data. We could evaluate it on any kind of measure. As I said, even measures coming from trackers that collect that type of sequential data. And implemented only for signaling networks. So that's something I was working on. And that's another method that I developed in the metabolic pattern analysis realm. So that's how I was trying to address that. So these are the limitations. I'm going to go quickly through the implementation details. So data processing was done. So this was implemented all in R. There is a package in R for it. It's not available yet. So trying to publish this work, and we publish the package at the same time. So data preprocessing was done using the athway PLM-R package, mostly metric data. The three-step function for that. Gene IDs were mapped to gene symbols using the respective annotation packages, and we used that for every real data set that we had. So this was for yeast, this was for fruit fly, for mouse, and for human. And gene expression at a specific time point was computed as the average of the replicates. So typically in the real data sets, we have multiple measurements for a time point. So how we use that in our data, we aggregate. And that's something we can work on too. Maybe we can gain more information if we look at the time points that of the samples that might have less noise. And now I'm going to take questions. I want to go to this method too, but I think this is enough for now, and I'll take questions. Thank you. So I'm really passionate about the topic in the sense preventive medicine. I like the idea of not allowing ourselves to get to the disease state and using every method that we can to prevent that. And these kind of detection methods, I think, help with that in the sense my dream is to have a method like this that collects data from the trackers that we have and tells you need to sleep more, you need to eat an apple, you need to do something, and that can help you stay healthier longer. So that's what this work is about. Yes.
You often don't have multiple time points on the same patient, but would you just suggest like you did with the liver where you take different pre-malignant states, try to analyze those, and use that as a proxy for time in essence, and pathologic progression?
Yeah. Yes, and also, so that would be the use case for cancer. And specifically, I have some slides at the end. I think I'm going to go. So by looking in the change interval, there. So we also looked in the change interval at the genes that change there, and that are on the pathway that we actually used for our analysis. The genes that were included in our analysis and genes that change during the change interval. We would think those are early markers of the progression towards disease. And we found genes that are of interest for hepatocellular carcinoma. They're not new genes, they're not new candidates, but it again gives us confidence that the method is going in the right direction. So that's a use case of the method. You would analyze the cancer stages, the data from cancer stages, and then when you identify the interval, you would look in that interval to see the genes that change. Because if you look from the beginning, from the first time point to the last time point, those genes might not change there. Because they might either plateau or revert after a point, and those might actually give you some insight to the disease. Then.
Do you have any idea about the computational requirements that your package would need? Given, for example, for every time point, you have, for example, one full gene expression set from cell line, and you have, for example, 10 time points. So how long and how resource intensive your process would be?
So all these analyses I've done on my laptop, the regular Mac, yeah, and it took minutes to tens of minutes, the most minutes, yeah.
Because
and some analysis, the ones for the simulation data where there were fewer genes and simpler networks, like they were almost instant.
Yes, so the reason I'm asking about this is although doing a regular time point sampling is not, and doing, for example, direct RNA sequencing is not possible at the moment, just because it takes a long time. But moving forward, the long read technology with Oxford Nanopore, they're actually inventing, and a lot of people are actually working on this, but moving forward, direct RNA sequencing is actually a very likely thing. And that too, within minutes of getting the patient sample. So
that
you can actually,
yeah.
I mean, if a program like this is integrated with that kind of sequencing method, it can actually predict patient outcomes right in a few hours as the patient state is changing from regular to disease. And something like this can be very effective in, for example, in ICU.
Yeah, and I'm thinking also not there, but it can help us devise better treatment, so we don't treat disease when it's not there, and we don't use toxic preventive measures. So I think it goes to more targeted, personalized medicine, and what you described is exactly that, like catering to the patient right there, right then. Yeah. Thank you. Yes.
Do you think this could handle diseases that have like mission and onset kind of on it?
So I think the fruit fly, the drunken fruit flies example shows us that they could. So maybe not as good as the other cases, but it could. It actually identified that there were transition back to that stage, and that's why we didn't have to metastate.